If you can take your seats, we're going to get started again momentarily.
Can probably go. Did you ride in? Did you have any near-death experiences? Sure.
Klymkowsky or Klymkowsky?
Yeah, Klymkowsky. It's phoneticized.
Okay, let's make a start. I hear a lot of stochastic stuff, a lot of noise. Okay, welcome back to the second part of our first session. And if the second part is even half as good as the first part, I think we're in for a really mind-stretching session here. And especially so, I'm really interested to find out more about what this first talk is about. So Mike Klymkowsky is talking about coping with the noisy nature of life in teaching and other philosophical ruminations. Mike, the floor's yours.
Thank you. Thanks to Larry for the invite. This will be at least, well, hopefully as 50% as interesting as the first two talks. And I'm a little nervous because I think my children are watching, which is the most, oh my God, the English major lawyer is in the front, so this could get scary. Okay, so, I'm gonna go backward. I want to start by giving you some of the intended take-home messages for this talk. First, it's worth recognizing that biological systems are inherently unpredictable. And they're stochastic, and that's a word we're going to come back to and define in multiple ways as we go on. The second is that what we are and what we do is influenced by, but not determined by, genes and environment. And I think this is the message of studying biological systems. It makes it a real difference between when you're thinking about chemical systems or physical systems, because this noisiness is built in. And luck and our response to it are important. There are good reasons to embrace free will over determinism, and we'll get to that toward the end. And this is not just a quixotic obsession of my own, coming from teaching and various molecular biology research projects in the past. Just the other day, there was a splash headline on the BBC about how genetics determines our life choices. There are many of these hooks sent out into the sea to catch unwary fish. But last year there was a talk from the author Karen Hardin on the genetic lottery, talking about how genetics influences people's social outcome, which I would say is a misfocusing of our attention. And recently she published a review on genetic determinism. Some phenotypes, such as Huntington's disease and other Mendelian disorders, are both genetically determined and 100% inheritable. We'll come back to that in a minute. So this idea of determinism and essentialism is underlying lots of them. The whole race debate is based on the idea that there's something different about different populations that's intrinsically essentialist, and that's just like silly. So, but sometimes genetic determinism is a great excuse. This is a great quote. For years, I tortured myself over not being more successful. This applies to me. Making more money, not giving up in between snacking. Now I know my failure doesn't stem from personal inadequacy, but from a lack of the required genetic wherewithal, but not so fast. Okay, so the first thing to keep in mind is this idea of genetic determinism is not supported by the data. Okay, so this is a review, same genetic mutation, different genetic phenotypes. 3 individuals can carry the same disease-causing mutation, and they will display different phenotypes. Some may display the disease, some may not. Penetrance is defined in genetics as the measure of proportion of individuals in a population who carry a disease-causing trait, allele, that express the disease phenotype. It's worth keeping in mind that diseases with incomplete penetrance, including Huntington's and breast cancer, are incompletely penetrant. So whether you express it or not is not deterministic, right? Just because it's there. And so this is a good review that was in the New York Review of Books about why biology is not destiny in response to this genetic lottery paper. First, disclaimers and caveats. I am not a philosopher or philosophy professor. I'm just a simple molecular biologist. Although I have ruminated on how Socrates would teach science, and I think it would be more effective than is commonly taught. And I make no claims about ultimate truth, because I don't believe science makes claims about ultimate truth. That's the domain of religion. Science is to figure out how things work and how to make things work the way you want them as much as you can. I recognize the challenge is to be comprehensible. A quote from Daniel Kahneman: if you care about being thought credible and intelligent, do not use complex language where simpler language will do. I'll try to do that. There are parts that could be, after the first 2 talks, I'm no longer afraid of overwhelming you. So. Basic working scientific assumptions are worth putting out right from the beginning: that the universe is natural and not supernatural or miraculous. It's things that can be understood, studied. Not everything is knowable. And this is something really important to recognize right up front. Okay? And hype and posturing about unknowable multiverses, the universe as a simulation, angels on the head of a pin, is actively anti-scientific. Okay? And I actually blame a large part of the physics community for going off on crazed tangents, talking about things that are unobservable, and essentially turning their back on the basis on which effective science is done. And maybe it contributes to the wacky world of QAnon and various people. So physicists should, I abandoned my Roman Catholic background some time ago, once they changed the Mass from Latin to English, and I could understand what people were saying. But I still regret not being able to go to confession on a regular basis. So, so this is really the story of a journey, which I started a few years ago, thinking about how to teach developmental biology, which is our last required course in molecular biology. And this is an incredibly tricky problem, and I came to this conclusion that a good focus would be on common social-cellular processes, how cells talk to each other. Because essentially most cells never exist independently. They're always in an environment, they're always communicating with one another. And it provides a context to consolidate and apply principles introduced in earlier courses. And many key developmental events are stochastic. So you may not have heard the word stochastic. It essentially means lawful, noisy, but lawful. And we're going to talk about that more. But the ubiquity of these processes in biological systems is rarely acknowledged and rarely taught. And that, I think, is a mistake, because it gives the illusion that biological systems are deterministic. And so it all starts with cells. This is an accurate representation of bacterial cells showing all the molecules in it. So these are bounded, continuous, non-equilibrium systems. They're based on selection-derived information that's stored in both DNA and cell structure. It's important to realize that all the information in DNA means nothing outside the context of the cell, and the cell itself is ancient. So every cell in your body has a history going back 3.5 billion years, an uninterrupted history. And all evidence suggests that we're all derived from essentially a single cell type, the last universal common ancestor. Cells differ in terms of the genes they contain and the proteins they encode, and that's what makes the different structures and processes possible. Now, there's a lot of buzz about AI. Larry was talking about it. I thought I'm going to make really cool graphics using DALL-E. Turns out it still has a little way to go. I'm not going to show you its picture of a 3D rendering of chromatin because it's incomprehensible. But at least we can see here that there's a strand here that sort of shouldn't be there. So you'll have to put up with my more pedestrian attempts. So an important thing to realize is that cells obey the laws, physical-chemical laws. Right? They don't violate any physical-chemical principle. That's one reason, aside from trying to weed out people who want to go to medical school, to require people to take chemistry. Right? And the energy of bond breaking for all reactions, whether they're covalent reactions or they're non-covalent, comes from thermal motion, except in the rare cases where it comes from absorption of light. Right? So it all comes from thermal motion. That's Brownian motion, which is inherently stochastic and unpredictable. Okay, when bonds form, energy is when bonds form, energy is released. It's transferred into the media. Enzymes, which are catalysts, and chaperones, which are essentially molecular chaperones, helping proteins achieve the right conformation, control reactions and interactions. But cellular processes are inherently noisy. So why are they noisy? Well, it's really quite simple. Cells are small. Okay? A typical chemical physical system consists of bazillions of a few types of molecules. Bazillions is a special word, which I'm extremely proud of at the moment. Each cell contains relatively small numbers of many different types of interacting molecules. You saw that in that picture of the cell. That only showed the macromolecules. You didn't see all the little ones. And they contain relatively small numbers of different types of interacting molecules. And most importantly, they contain 1 or 2 copies of each gene. Right? So this is as small as you can get. And what do we mean exactly by noise? So there's a lot of confusion in the literature. Words are being used all over the place in sort of sloppy ways. Words matter at the end of the day because they have implications. Noisy does not mean random. Okay? Random implies there's no natural cause for the event. It's a supernatural event. It's essentially a miracle. Okay? It displays no pattern. Here's a classic example early on. Stochastic means individual events are totally unpredictable. Okay? We can't tell when something's going to happen. But when we have a large enough population, the outcome becomes predictable. So a classic example is the throwing of a dice, a pair of dice. For any one throw, the outcome is unpredictable. But as the number of throws increases, the overall outcome becomes predictable. So if it's a fair dice, as the number of throws increases toward infinity, I will get equal numbers of each side of the dice. Now, the important thing to keep in mind here is often called the gambler's fallacy, which is associated, I keep forgetting that I'm living in Colorado. The gambler's fallacy is if I throw 6 throws of a dice and they come up 6, then the probability of the next throw will be something else. No, the probability is always the same. Each throw is independent. Okay? So each throw is independent, not influenced by the next event. The same is with radioisotope decay, which is probably a more familiar example. When any particular unstable atom will decay is unknowable. It's unpredictable, it's unknowable. It could happen in the next moment. It could happen a billion years from now. But if you consider bazillions of atoms, the time at which 50% have decayed is extremely accurately predictable. Okay? But generally, the difference between biology and chemistry and physics is generally it doesn't matter whether it decays or not in a particular moment. It doesn't have any real significance. I took out a series of slides on Schrodinger's cat, because I didn't want to discuss the quantum nature of reality because I don't understand it. So, but this is not the case in biological systems, okay? In a biological system, a stochastic event can matter big time. So the obvious example is your DNA in one cell gets hit by a cosmic ray, and it mutates, and the outcome is a mutation. Same thing can occur if it interacts with a chemical, and can lead to cancer, a developmental defect, novel behavior, death of the cell. Right? So this is an event that occurs because you did one little thing in a cell. Now, normally, there are plenty of feedback and moderating effects that help fix it, so this mutation could be repaired or not, but it could give rise to cancer. So this one event, which is unpredictable, is significant. And that really is what makes biological systems so different. And in fact, this level of stochasticity goes all the way up from the molecular to the neurological level. Okay, beyond mutation, how molecules interact is stochastic. The interactions are based on surface charges and molecular shape. Molecular movements are driven by unpredictable and rarely illustrated collisions. I've been doing a lot of working on textbooks, and when you look at a model of how like the ribosome works, all the tRNA molecules know exactly where they're going, and you don't see the fact that only 1 out of 20 or 30 collisions with the ribosome is functional, right? Because it looks too stupid to run a system that way, right? But in fact, that's how it works, right? So once we get that interaction, okay, boop. Now the kinetic energy that breaks these bonds is delivered through collisions with surrounding molecules. That's Brownian motion again, right? So how long two molecules will stay coupled is stochastic. It's not predictable, okay? How long they will remain together, if I look at bazillions of complexes, is predictable. Right? That's the key of stochastic processes. Stochasticity can be a difficult concept, often not taught explicitly or effectively. And students and most people, sometimes the overlap between the two groups is still subject to some conjectures. Students favor active processes. Okay? They think that why molecules come apart is because they should come apart. Okay? The system needs them to come apart. And so this is a question from the biology concept inventory, which I built with Kathy Garvin-Doxis some time ago. These are questions that are based on students' responses to specific queries. And so the difference between this and a classic multiple choice test is because the wrong answers, which are called distractors, are things that students actually think, right? So if you give a multiple choice test where the student doesn't think the wrong answers could possibly be right, then you overestimate what students know. That's the problem with classical multiple choice tests. But what you can see here is like, well, once two molecules bind together, how could they come back apart again? The distractors are, they would have to bind to another molecule. That's an active process. The complex would need to be degraded. The chemical reaction must change the structure of one of the molecules. That's the most popular one. And collisions could knock them apart. Well, it's always collisions that knock them apart. It's nothing else. There's nothing else that's going on in the system, okay? It's just collisions. So you can look at this. This is the percentage of students who answer this. It doesn't change much. In fact, it gets worse in the course of a conventional molecular biology course at a highly prestigious university in Switzerland. And it turns out that we are not effectively getting this idea across. Right? And part of it comes in the context of thinking about how people are taught chemistry. Often in chemistry courses, people are introduced to enthalpy and entropy and delta G, which are extremely abstract ideas and are often measured and are essentially always measured in bulk, right? So the idea that the binding interaction is basically a function of how long it takes for thermal collisions to knock the molecules apart is a more mechanistic view, more accessible view, I think. So stochastic gene expression is everywhere, and so I do a flash from the past for Larry, because he will appreciate it. This is classical molecular genetics. So lactose, which is mammalian milk sugar, is energetically expensive for bacteria to use. It requires the synthesis of lactose-specific enzymes to let lactose in the cell and then to metabolize it. But a bacteria may never encounter lactose in their environment, right? So the question is what to do. If lactose is there, I want to be able to use it, but I don't want to be making all these expensive enzymes if it's not there, because I could be using that energy for something better. So how does one sense the presence of lactose? Well, when a cell needs energy, it can ask whether lactose is present. So how do cells ask questions about their environment? They turn on genes needed to sense and utilize lactose or other components of the environment. And they generally do this by what's a burst of gene expression. And so we can look at this in a population of bacteria and follow the expression of specific gene associated with utilizing lactose. And what we find is that the genes come on, essentially stochastically. If there's no lactose, the genes turn off again. Okay? So there's no need to express this expensive lac sensing system all the time. It's energetically expensive. But what do I do if lactose is present? So the genes required to sense lactose remember, they're turning on, right? And if lactose is present, the population as a whole will turn on those genes and utilize lactose. And what's going on at the single cell level is, again, the cells are expressing the genes for lactose utilization stochastically, but if lactose is present, they stay on. Now, how does this work? So this is my most complicated slide. So the lac operon, which encodes these genes, has a positive regulatory site. It has a negative regulatory site, which is bound to a lac repressor, which is always expressed. But it's expressed at very low levels, so it's always being knocked on and off. Remember, it's recognizing specific sequences in the DNA, which are short. There's another few million base pairs of DNA around, so it binds non-specifically at weak affinity to those sites. So often this molecule will come off and it'll take a while for it to go back on. But it doesn't matter because the positive regulator is not there. When other easily digestible energy sources are present, the gene is off. Why? Because the protein is adopted a configuration where its DNA binding domains are not visible. They're hidden. Okay? So it's inactive, so it doesn't matter. Repressor comes off, doesn't matter because the gene's not active. In a starving cell, on the other hand, a signal is generated, cyclic AMP is made. It's an allosteric effector. It binds to the cap protein. It activates it. It causes its configuration to change. Its DNA binding helices become visible. The DNA, and it binds to the cap site. It recruits the polymerase. When the repressor comes off, the polymerase can transcribe the gene. When those proteins involved in lactose metabolism are present, lactose enters, it's metabolized to allolactone. This is an allosteric effector of the repressor. It binds to the repressor and stays off. So that's why it stays off, the gene stays on when lactose is present. And so a classic example of this, and something that I used a lot in class, is a study by Elowitz in 2002. So you take green fluorescent protein, and it can be mutated to make cyan fluorescent protein, yellow fluorescent protein. So you can make it's the amazing world of fluorescent proteins. You take a plasmid encoding a piece of DNA, encoding these fluorescent proteins with independent regulatory regions. You introduce them into genetically identical cells, and you're growing them in a homogeneous environment, and this is the plasmid. It's actually even more sophisticated than it may look because Elowitz put the two genes equally far away from the origin of replication, so DNA replication can't affect them differentially. So they're basically in identical molecular environments. And you look at the cells, and what you see is pretty straight up. You see some cells that are not expressing. You see some cells that are expressing yellow, some cells that are expressing green, and when both proteins are expressed in the same cell, it's red. So you see that in this genetically identical environment, there's stochastic variation. It's unavoidable. The only way you could avoid it is by controlling, and the way cells do try to control stochastic processes, is they change the levels of proteins so that they suppress it. So if you want the repressor, if you want the lac operon to never come on, you increase from 10 copies of the repressor per cell to 1,000 copies, and it'll never turn on, and the cell will never be able to respond to lactose. So you can tune the system for various levels of noise that you like. Whoop. Okay, stochastic choices are critical for social systems. This is the link into developmental biology. Individual unicellular organisms can cooperate with one another to produce structures and behaviors that they could not form on their own, right? So the classic example is the slime mold dictyostelium, but there are many of these examples. And in dictyostelium, what's going on is that, whoop, I don't want to do that. I want to do this. Whoop. Oh yeah, see, I've now discovered the button problem. If there's lots of bacteria, which the amoeba eat, these are eukaryotic cells, so they'll eat it, they're happy, they're growing in soil, they're eating the bacteria, all's good, they're independent organisms, right? What happens is, if they start to starve, they undergo this transition where they go from unicellular independent to multicellular cooperative. So they release signals, they talk to each other, they aggregate, they form what's called a slug, which can migrate around, come to the top of the soil, because what they're going to do next is they're going to essentially differentiate. They undergo this set of processes where they form this long stalk and a head. In the head, there are the spores that are going to be released into the air and move away from this barren area. The stalk is made of cells that sacrifice themselves. They die to make that stalk. Okay? Cooperation involves sacrifice. We all know that. If you're in a room with extremely rude or boring people, you have to be able to control your emotions so you can continue to live with them and not kill each other in some horrible way. So this is altruistic behavior. Boop. This is an example of what it looks like as this stalk is lifting the spores out of the soil. Sacrifice best achieved through stochastic choices, because if it was genetic, if it was controllable, all cells would want to become spores. Once all cells become spores, right, once there are no electricians, or plumbers, then your house collapses, right? So if everybody tries to become the spore, you defeat the purpose of the cooperation. Cooperative behavior and stochastic choices are everywhere in biology. You think of this in the early mammalian embryo. I had this slide checked by John von Blerckum, a famous in vitro fertilization pioneer. In this case, the cells divide. They form a compact, and the cells that enter the inner region of the embryo, which form the embryo proper, are selected stochastically from the surface cells. And there's a lot to be said about this, which we're not going to say. Which cells become the germline, that is, give rise to sperm or eggs, and which become the soma, which support the germline. That's again stochastic. Stochastic effects influence the development of essentially all systems in the body, from the immune system to the nervous system. So here's an example of thinking about what a stochastic event does. and this is from a paper by Saka and Smith, which I think is really a nice teaching opportunity. So this is a simple gene network where a signaling molecule is positively regulating the expression of these two genes, which form various interaction negative feedback loops with one another. This is like extremely similar to essentially all regulatory circuits in every cell for every process. Whether A or B is expressed is influenced by multiple parameters. It can be one or the other. Which turns on first leads to different outcomes. Typically, each regulates many downstream genes. A typical transcription factor in a eukaryotic cell will regulate hundreds to thousands of genes. And that pattern will lead to different, often irreversible changes in cell fate and behavior. So that simple decision of whether I'm going A, B, and who binds to which site first is stochastic in most cases. We can think about noisy neural networks. This is a classic example. One of the first examples of looking at noise was watching the opening of ion channels in neuronal membranes. This is an example of a neural net. The opening and closing of channels is stochastic, right? It's driven by thermal motion, which knock into the protein and cause it to open, or it closes, right? Again, the same way. You can control the level of stochasticity because the amount of energy that's required will depend on the resting potential of the neuron. The closer I get to the threshold, the less energy is required to open it up. Now, I was thinking about this and what would be a really sort of engaging kind of study. And so this is a paper that was published, I think, in 2019 in eLife, and it's an intriguing study. You may have heard of these experiments in which the readiness potential, which you can measure, precedes a subject's conscious decision of what they're going to do. And this was used as an example to argue against the idea of free will. Right? You were not actually controlling the decision that's already made before you knew you made it. Okay? But it's important to note that previous work focused on arbitrary decisions. These were purposeless, unreasoned, and without consequence. So these authors tested the idea of whether this was true for a deliberate consequential decision, where you had to think about what you were going to do first. So they also confirmed that this readiness potential is present for arbitrary tasks. It was absent when you have this, when you're you're actually thinking about what you're doing and it matters. Okay? This suggests that the RP represents the accumulation of noisy random fluctuations that drive arbitrary choice. But different neural mechanisms, maybe consciousness, underlie deliberate decisions. Certainly, feedback within the nervous system that's involved in thinking is playing a role. So part of this is, and comes back to this idea of the lac operand, how do I know the state of my internal state if I'm a cell? How do I know what's going on outside of me? I'm making models, right? One model is the lac operand. I'm going to turn on the lac operand for a burst. If I'm going to guess that maybe lactose is there, I might need it. If it's not there, I turn it off. So at various levels, cellular and organismic, models are rapidly constructed and revised. And this is sort of the basis of Kahneman's thinking fast and slow. This happens to me a lot. I'm walking along. I think I see a mountain lion trying to jump on me. My immediate professor, I'm ready for that mountain lion. And then I look and say, oh, it's a McDonald's hamburger bag. So you're constantly making models of the environment and then checking them. Right? So it's really important to have a model fast because you're not sitting there waiting to decide what's going on. And it's important to realize we're prone to overestimate how much we understand about the world and underestimate the role of chance in events. So there's a higher level of stochastic processes which you may already have known about. One is, the classic is X in activation. This is in XX females. the maternal or paternal X chromosome is silenced, and that's to balance expression, because males have only one X, and while males and females are arguably different, they're not that different. And so you're balancing that X. And that process of X inactivation in females occurs early in development, and it's stochastic. I almost said random. So it's stochastic. It's hard not to say it, right? And it gives rise to clones within the organism. So a female is a combination of clones in which X is expressed and which isn't, which other one is expressed. And this is, of course, the reason why calico cats are the way they are. Now, there are unexpected stochastic processes that are being revealed by new technologies, particularly single-cell RNA sequencing, which allows us to see at a cell-by-cell level which genes are being expressed in a particular cell. And that's monoallelic gene expression. In this case, in a particular cell, both alleles could be expressed, one could be expressed, the other could be expressed. If they are different alleles, different types of the gene, you can get different phenotypes in that clone. Okay, more autosomal genes are subject to monoallelic expression than are genes on the X. So this has potential of a big effect, generate clones that can compete with one another and interact with one another. To give you an example of this, I thought I'd use this one, which is stochastic monoallelic expression is found in the gene FOXP2, which controls language. Mutations in FOXP2 lead to verbal dyspraxia, which is a profound speech and language deficit. So monoallelic expression of FOXP2 can produce multiple competing clones, brain regions with normal, reduced, or mutant. So we're not even expecting the same genotype in different regions of the brain, essentially, the same functional genotype. So when we think about the brain, there are regions that are associated with reading words, hearing words, thinking words, speaking words. This is one reason I always ask students to read and I have the computer always read things to me aloud, because you use different parts of the brain to process language in different ways, right? So something that can sound really good to you when you're talking to yourself can sound really stupid when it's said out loud. And so you can see I'm not listening to myself for a while. So, so you can think about Wernicke's aphasia. Let's imagine that we had a monoallelic expression of a mutant allele in a certain region. All of a sudden, you can impact how the brain is constructing speech. Visualizing the stochastic behavior of cells has revealed lots of this variation. Here's a classic example. This is cells from a xenopus embryo, which are, each dot is an individual cell, and you're seeing a distribution that reflects the patterns of gene expression. Cells of the same type don't express the same levels of each gene. Okay, so they vary with one another. And in some cases, you have cells, here's some red cells over here, they should be here, they're over here. So you see all kinds of variation, which the organism has to control, one way or the other. Something that's become very interesting lately is the fact that there are cooperative effects between cells, so that eccentric cells can be induced to die or migrate if they're surrounded by normal cells. Right? So this is called cellular competition, which was not expected before. I mean, there were just hints of it before, but this is clearly one way of controlling stochastic effects. So stochastic effects cannot be avoided and can be useful. So this is a summary of 30 years of inbreeding experiments in shared environments, and it eliminated only 20 to 30% of the observed variance between organisms in a particular phenotype. 70–80% was referred to as intangible. This is a very nice review by Kyle Honegger and Benjamin de Bivort, in which they refer to these papers. If you're interested more, this is a really good review. Genetically identical pea aphids reared in the same environment show individual variation in their startle response. So you kick the plant, and some of them stick on the plant, some of them jump off. You can see the value of this right away. If you're a squirrel and you always jump to the right, when a fox is chasing you, pretty soon there are no more squirrels, right? If where you're going to jump is unpredictable, then the predator has to compensate, right? So variation is not a defect, it's a feature of biological systems. So the conclusions. So from this original idea of that you're determined by heredity and environment, what we are and what we do is influenced by, but not determined by, the environment. We can think of it more along this way. Given their ubiquity, stochastic processes should be introduced to students early so they can understand how these processes work. And my first act of free will is to believe in free will, according to William James. So thank you for your attention. The daughter liked it. Life is good. Life is good.
Mike, you want to sit or stand? I don't mind.
I'll sit.
I've got a first question online, though. It's from one of your kids. It says, I crashed the car stochastically? I don't know what that's about, but...
Me neither.
Mike, enormously enjoyed your outline and the logic that you stepped through, but I got stuck in the beginning and stuck in the end. You started with the statement, free will, if you lack free will, equates determinism.
Well, so I completely get this idea. So my view is, if something is not predictable, what does it matter whether it's an arbitrary choice in a sense. I mean, if we believe in a naturalistic system, something must be causing something, right? Even if it's not measurable, but it's not predictable. So the negative effects of denying free will seem to me to be to sort of overwhelm the positive effect is assuming that you have some responsibility for what you do. But they're both philosophical positions, as far as I can tell.
Yeah, they are. I'm trying to get to understand your philosophical position. I think we're going to do this over a glass of wine.
Sounds good. I mean, my assumption is I have to take some responsibility for my tragic errors in the past. Even though there may be a good reason why I misbehaved.
Hi, I wanted to first understand, just make sure I understand your definition of stochastic processes well, because I think that's fundamental. Where I think, if I understood correctly, it means a process that is governed by fundamental rules, but it's almost like too noisy for us to understand how those rules are in action?
Essentially, right? So if I think about Brownian motion, so here's the issue. If I think about Brownian motion, how would I predict what's going to happen? I'd have to know the mass and momentum of every particle in the system. That's unknowable because measuring it will distort the system. It won't be the same system when I'm done measuring it as it was before. So I can't measure it, but I can assume that all the particles have mass and momentum and they're running into each other, okay? So the key of it's a sort of weird idea that while individual events are unpredictable, in large groups, they become predictable.
Yeah, so maybe then I can
And the only reason biological systems break this is because there are no large groups. I'm not averaging every cell together to get a common cell behavior. The behavior of individual cells matters. If I get a tumor in my liver, then it only mattered that I got that one event occurred.
Yeah, but I guess maybe just then the follow-on to that, because you give the example of the fair dice at the very beginning. Can any human in reality ever prove that a dice is fair and it really will end up with equal probabilities of 1 through 6?
I think if you get up to a bazillion, you can be pretty confident.
But you're not. You're making an assumption. You're making an assumption. Case I'll say on a faith, right? Because it is impossible. At least I think it's impossible to prove the existence of a fair dice in reality. Yes or no?
I would say that. So my view of this is science. Again, science is not about truth. Is what you need to know to predict the behavior of the system. If I throw this dice a million times and the response comes out equal for each face, I can make a working hypothesis that it's a fair dice, and I don't care about the ultimate reality of the dice. The ultimate reality of the dice is unknowable to me. I'm just, I'm just an advanced, I'll get rid of the advanced. I'm just a hominid, and I'm just a modified monkey and the primate. And but this presumption that I can make these working conclusions and they work, and I can do what I want to do, is demonstrated all the time.
Time for one more.
Yeah, I just wanted to go back to something you said at the beginning. You're trying to teach students in a good way about thermal dissociation of complexes, their stochastic events, but actually your answers 1, 2, and 4 were also correct in many cases in biology.
But the reality is the only thing that knocks two molecules apart is thermal motion.
That's completely wrong.
No.
Many examples where a signaling pathway comes in,
We'll change the affinity.
complex.
We'll change the affinity of the two molecules for one another,
It's
but
strand
they will come
displacement.
apart because they're knocked apart by thermal motion.
No, come on, it's caused by, in those 3 cases, by directed purposeful events of the cell. I think you're teaching students that.
I'm
Okay,
willing
we'd
to
like to
brought
talk
up
about this some
my
more.
charges.
We love a bit of controversy, though. Thanks so much, Mike. That was really thought-provoking. And so in our last session our last talk for this session,
Yes, please.
while we're disconnecting, we're disconnected. We now have Alejandro